Could we test whether airway alkalinization in that subpopulation would be an effective targeted therapy? Just an example. And there are many, many. We could go on. We could talk about lipoxins. But what we have to do is define the phenotype. So when the person who walks into clinic with low GSNO, with increased GSNO reductase activity, for example, comes in, we know what that person looks like enough to test for it. So the last example I'm going to go to is to spend probably the rest of the time on GSNO reductase activity, and then we'll have time for questions. So this is always a little bit of a confusing example, and so I want to spend some time on it. This is the classic paradigm regarding nitric oxide synthase that so many of you know, and it seems like I'm shifting gears here, but this will come back to talking about severe asthma. So classically, we would say that NOS creates nitric oxide, which has cytotoxic effects or activates guanylate cyclase-dependent targets. But I want to broaden your view of the effects of NOS a little bit, and this turns out, I think, to be quite relevant to asthma. So here's what I just showed you. But in fact, NOS can also make an oxidized form of nitric oxide, which is essentially a nitrosonium bound to other species, in this case a thiolate, that can store and transmit the bioactivity of NOS in a lot of important ways. And this kind of signaling can be regulated by transnitrosation reactions, by localization, by degradation of these nitrosothiol species, or NO plus bound to other species that we'll talk about. This can cause intracellular cyclic G independent effects, in addition, by the way, to activating cyclic G by causing more guanylate cyclase activity. These can be stored in vesicles. These nitrosothials fused and export in the form of S-nitroso coenzyme A, S-nitrosocysteine, and they can also be stored and synthesized in NOS independent fashion. And here's one example that's of some relevance to asthma, just to show you how this kind of signaling is studied. This is from the Snyder lab in Baltimore, looking at COX-2 activity. So it turns out that COX-2 activity is regulated by inducible nitric oxide synthase, and in fact, if you inhibit iNOS, you decrease the readout of COX-2, and this is in macrophage cell lines, in the context of PGE2 production. Similarly, if you look at the NOS knockout, the amount of PGE2 formed in response to LPS is high in the wild type, but quite low in the knockout. If you look at S-nitrosylated COX-2, there's a specific cysteine on COX-2 that's modified by activation of iNOS by LPS. S-nitrosylated COX-2 is not at all evident in the iNOS knockout mouse, even with LPS and interferon gamma stimulation. And it turns out the bottom line of a complicated story is that activation of iNOS, S-nitrosylates COX-2, activating COX-2 and making PGE2. This is the kind of paradigm that we're particularly interested in studying in our lab as it relates to a variety of things, one of which being asthma. And indeed, as the assays have gotten a little bit better for nitrosylated proteins, the number of publications has increased fairly exponentially. This was me and Stamler, and this is when there was a raging controversy about hemoglobin and the assays and so forth. But I think people realize now that was kind of ridiculous, and there's been a tremendous increase. All right, so how do those redox reactions occur? Let me just, so when you think about asthma and NOS, you think about iNOS. This is a picture of eNOS in pseudostratified airway epithelial cells grown at air-fluid interface. So this is a standard UNC model. So we've grown these to form a ciliated layer at the top, and you're looking at the z-axis, and the red immunostaining here is for eNOS. You can see, as Roger Johns has published before, that eNOS is localized right at the very top of the pseudostratified epithelium, and we see this also in immunohistochemistry ex vivo in human airway. And eNOS is activated, it turns out, by cyclic compressive stress, by moving cilia, by moving air across these cultures, and it makes nitrosothiols, and actually in a NOS inhibitable way. So that if you subject these air fluid interface cultures to breathing, as has been pioneered here at Harvard School of Public Health and elsewhere, you make a lot more nitrosothiols than in controls. There's an interesting electron acceptor protein underneath the eNOS, and in some cases co-localized with it. I can't go into that in too much detail at this point, but it should come out soon. Sorry, Bruce left because this is talking about the assessment of nitrosothiols in the airway of humans in vivo, and this is from Bruce's bronchoscopy. This was done here 20 years ago. This was Catherine King's first bronchoscopy done here at the Brigham, and I was the subject. And it was all legit. It was Jeff Drazen's protocol. But there were a lot of people who wanted macrophages, so there was a long line. Bruce was at it for quite a long time. And once I recovered, I was surprised at the CNS effects of all that lidocaine. He said he didn't give me anything, but I was like, whoa, what just happened?
Wasn't it a consent form?
No, I don't think so. But once I fully recovered, I took my BAL and went over to the Perlmutter and spun it down, and did this reaction on the old Shimadzu over there, which is basically a nitrite assay with and without mercuric chloride. And this difference between with and without mercuric chloride turned out to be primarily by HPLC S-Nitrosoglutathione. Glutathione is very abundant in the airway lining fluid, and this S-nitrosylation modification of glutathione also turned out to be quite abundant. This was about 1 micromolar concentration, and we subsequently did a lot of subjects. And it turns out that if you took human airway smooth muscle cells, and these, or tissue, excuse me, these were kindly given to me by a sugar baker, usually late at night, and contract them with methacholine or histamine or leukotriene, then you could expose them to increasing concentrations of theophylline or isoproterenol that would cause them to relax. This middle line here is S-nitrosoglutathione, which caused in human airways a cyclic G independent relaxation of the human airway smooth muscle. And I bring to your attention that the IC50 is pretty close to this concentration that's normally in my airways. So this was an endogenous molecule that relaxed human airway smooth muscle and seemed to be of interest with regard to asthma, sort of an endogenous bronchodilator. The cyclic G independent effect has taken a long time to figure out, and we still haven't got it entirely, but one of the things is that it modifies specific redox-sensitive cysteines on the myosin head in a stereoselective way. That is to say that S-nitrosyl L-cysteine is active in interfering with actin-myosin interactions at the myosin head, while the D isomer is not. So this is not a nitric oxide effect, and it's not a cyclic G effect. If you UV treat the stenosis before you do it and evolve off nitric oxide in situ, you don't get any effect. It's a transnitrosation reaction causing modification of the specific part of the myosin head of interest. And so there is a direct effect of nitrosothiols on actin-myosin interaction in smooth muscle and also in cardiac muscle, but it turns out that also there are a variety of other interactions, and I won't belabor this too much, but with regard to asthma, this is work that was published a few years ago in Cell, talking about the effect of S-nitrosoglutathione on beta-2 receptor expression. There is a mouse that is deficient in the enzyme that breaks down this molecule. We talked about synthesis, but breaking down the molecule is done primarily by an enzyme called S-nitrosoglutathione reductase. And just walking you through this, these are heart data here. So if you look at isoproterenol stimulation of the heart, NOS inhibition inhibits the effect of isoproterenol on the heart. It also inhibits beta-2 expression. But so isoproterenol, this is a long exposure to isoproterenol of heart muscle, will decrease beta-2 expression, tachyphylaxis basically, which I think you all know. But GSNO rescues that in the presence of isoproterenol. In fact, there's something of an increase in beta-2 expression with isoproterenol that I'm not sure I fully understand. In the GSNO knockout mouse, there is some effect to improve beta-2, not having the enzyme that breaks down GSNO improves beta-2 expression in the heart, but even more in the lung. And this is a cartoon of the modification of G-protein coupled receptor kinase 2, GRK2, which by eNOS-dependent nitrosylation, and remember eNOS is in the airway epithelium as it is in the smooth muscle, and the endothelium, and that prevents phosphorylation of the beta-2 receptor, preventing beta-arrestin complex, and preventing internalization and loss of the beta-2 receptor in response to ligand binding, which is a long story made relatively short. But this may be another cyclic G independent mechanism by which nitrosothiols augment human airway smooth muscle. Remains to be proven for certain. In the context of all of that, well, not knowing anything about mechanism, but in the context of knowing that it was a smooth muscle relaxant and that exhaled nitric oxide levels are very high in asthma, we kind of expected to find very high levels of nitrosothiols in asthma. And what we found was quite the opposite. So these were 8 intubated children in San Diego with asthmatic respiratory failure compared to other children in the operating room for routine herniorrhaphies and so forth, non-thoracic surgeries. And these levels were, somewhat close to where mine was, around a micromolar. And most of the asthmatic children had undetectable nitrosothiols levels in general, and nitrosoglutathione in particular, in their airways. So bronchodilator deficiency and asthmatic respiratory failure. And in fact, it turns out that the reason was not so much that less of this was being made, but more of it was being broken down. And so if you took 1.5 micromolar GSNO and spiked that into one of these fluids, half of it was gone at 10 minutes. Where in one of these fluids, most of it was still there after 10 minutes. So we subsequently spent a lot of time looking for the enzyme that broke down GSNO in these infants. We isolated it in a guinea pig and predicted the KM. And it turned out that this enzyme was S-Nitrosoglutathione reductase. Loretta Quay and Jonathan Stamler's lab went on to, with Li Min Lu, to knock this out in a mouse and show that in an ovalbumin sensitized model exposed to increasing concentrations of methacholine. So an old-fashioned sort of mouse model of asthma, for what that's worth. That the mouse that did not have S-nitrosoglutathione reductase was protected from ovalbumin-induced increase in airway resistance in response to methacholine. And this is without sensitization